From the Biotechnology Life Lab Longwood, this is the Pulse Podcast. I'm one of your co-hosts, Maldika Dua.
And I'm your other co-host, Tanner Baldwin.
Together, we dive deep into biotech stories and feature biotech leaders. This podcast is brought to you by the Griffin GSAS Harvard Biotech Club, a trainee-led student organization which is on a mission to bridge the gap between academia and industry. The podcast is also supported by the Biotechnology Life Lab, a Harvard affiliate incubator aimed at accelerating biotech startups that have an impact on human health. Today's guest is Dr. Tim Harris. Tim is a molecular biologist, biochemist, and geneticist. He is also a science and business leader with over 40 years of experience in a range of molecular research areas. Tim is currently an operating partner at SV Health Investors, and outside of SV, he holds a seat on the board of directors of Phenom TX in Edinburgh and is a visiting professor at Columbia University.
Before joining SV, Tim served in several senior management positions, including Executive VP of Corporate Development at Repertoire Immune Medicines, Executive VP of R&D at Bioverativ, now part of Sanofi-Genzyme, the Senior VP for Precision Medicine at Biogen, which followed his role as Senior VP of Translational Medicine also at Biogen, and Senior Vice President of R&D at Sequana, Axys. Tim also served as Chief Technology Officer and Director of the Advanced Technology Program at SAIC-Frederick in Maryland, currently known as Frederick National Laboratory, which operates the National Cancer Institute's leading center for cancer and AIDS research. In 1999, Tim founded SGX Pharmaceuticals. He built SGX to more than 130 employees, raised $85 million in capital, and generated more than $20 million in revenue during his 6-year tenure as CEO before it was sold to Eli Lilly. In 2024, Tim published his well-regarded memoir, In Pursuit of Unicorns, a journey through 50 years of biotechnology. With first-hand experience of biotech's evolution over decades, the book highlights the most impactful advances in biotech and gives a compelling insider view of the breakthroughs that have shaped modern biotechnology. John Maraganore, former CEO of Alnylam Pharmaceuticals and a previous guest of The Pulse, has praised it as a new Bible for the history of biotechnology. It's our honor to have Tim on the show. Welcome, Tim.
Thank you for having me. I appreciate being here.
So we went through a lot, and you've had, as I mentioned before, you've had such an illustrious career, but we wanted to go back to the beginning. And to start, we wanted to ask about your upbringing and what early in your life actually drew you and sparked your interest in science altogether.
Well, when you get brought up in a household where your father is a chemist and there are copies of Nature on the kitchen table, then it's not very difficult to wonder why somebody would be excited about reading Nature. So, not that I understood it or read it when I was a kid, but it was there, and he was very much into cancer research. He was a chemist by training, so that was really the beginning. And then one memorable afternoon, I was outside, and this old guy was walking up the road, and it turned out, because I asked my dad who it was, and he said it was Sir Robert Robinson, who was a Nobel Prize winner for discovering the structure of various natural products, and worked in Oxford and at Shell in the UK and lived up the road. And I was kind of wondered why somebody would want to do that as a living. So that, again, sparked my interest in science in general. And, when I was thinking about going to university, I'd already done science A-levels, as you do in England, because you have to choose whether you want to do arts or sciences quite early on, and I chose sciences. And when I was deciding what to do, and I thought I quite liked biology, and I thought biochemistry would be a good subject to do because it was a mixture of chemistry and biology.
So what was your actual focus in your PhD, and how did that afterwards lead you towards the biotech sector, and what kind of thinking actually helped you in your journey throughout the rest 50 years of your life?
Yeah, well, after doing a biochemistry degree in Birmingham, I went up the road, literally, to the virology department, which was run by a guy called Peter Wilde, and they had an interest in herpes viruses, and my PhD was looking at messenger RNA synthesis in herpes virus infected cells, which gave me an interest in messenger RNA right from the beginning. And as you know, messenger RNA is a crucial molecule discovered in part by Sidney Brenner as being the intermediate between DNA and protein, way back when they were discovering these things. And I thought it was one of the most exciting molecules that you could possibly study because of its role in between DNA and protein. And so herpes viruses and viruses in general, as you know, are a good way of getting at messenger RNAs. And I left, having finished my PhD, to go down to the Animal Virus Research Institute, as it was then in Pirbright in Surrey, to work on Sequana viruses, foot and mouth disease virus in particular, but poliovirus as well. And again, as you know, that's a positive strand virus, so that's a messenger RNA in itself. And then I came to the US for a year to work on polio, came back to the UK, and the whole recombinant DNA thing was just starting in 1978. And I cloned various bits of foot and mouth disease virus RNA as DNA copies and other viruses as well. And we, in fact, in a petri dish with 280 clones, we got nearly 90% of the genome in one cloning experiment, because it was a pure RNA, so you'd expect to get that if you could get the clones to clone, which we did. And the only bit we were missing was the bit at the 5' end, which turned out to be just before a poly-C tract, which was difficult to read through, which was no surprise why we didn't get full-length clones. But we cloned we managed to find the other bit as well, and then you got a full-length copy of the RNA as DNA, and you can start to do interesting experiments, which is what we did. So that was my interest in cloning. And, once I've been at Pirbright for 3 or 4 years, Celltech was on the horizon in November 1980. I read a Nature article about the formation of Celltech, and I'd already been collaborating with the guys who started Celltech. So I thought, well, what better thing to do than to go to a new company in the biotech space, just starting to run the cDNA cloning lab, which is what I did for several years.
Interesting.
Really, really good fun.
Yeah, so can you tell me a little bit more about that? You started in virology, then you were one of the first molecular biologists at Celltech. And tell us a little bit about that experience, and specifically what led you to make that transition from academia to industry, especially when it was back in the day, it was less common.
Not so difficult when you double your salary and you get a company car, right? I mean, and I had 2 kids. Well, I had 1 kid and 1 on the way. My second daughter was the first Celltech baby in 1981 when I joined. And so pretty exciting, actually, altogether. So, yeah, it wasn't really about the money. It was about the applied nature of the job that I would be doing. In other words, we were making things that I thought might be important, tissue plasminogen activator being one of the projects we were working on. And it kind of appealed to me that I was cloning genes with the purpose of making an enzyme which you might use as a drug. I thought that utility was important to me. I didn't just want to find out more and more about less and less, if you like, from an academic perspective. I wanted to apply what I'd learned. So, if you like, the application of science, the application of technology has always been something that I've been extremely interested in. And if you like, that's the reason why I joined the dark side.
Right.
It's not the darkest of the dark sides, by the way. I'm now on the darkest of the dark sides. But the industry was once the dark side. It really isn't anymore. It's a really good place to do applied science. If you want to make things that might help people, then it's a great place to go.
Yeah, you mentioned for maybe a lot of practical reasons that transition maybe wasn't all that difficult, and it seemed like you were really enjoying what you were working on, which makes a transition like that a lot easier. But did you meet any resistance from your colleagues? Like you said, moving from academia to what was once sort of the dark side of industry is a lot more common these days, but back then maybe not so much.
I don't think there was any resistance, actually. I think people were a bit intrigued because it was a new startup. Celltech had only just started, and I don't know, people were not was no jealousy involved as far as I could make out. It was just, well, it's a different path you want to go down. And quite a few people joined. And don't forget, I wasn't really working in academia. I was working for the government as a government research scientist. So if you like, that's a second level down. It wasn't pure academia. It was, we were trying to find a new foot and mouth vaccine. We were trying to find out more about how the virus worked so we could do things about cattle that were infected. So there was always an applied nature to that work. So it wasn't that much of a jump from that. But as you say, these days, moving from academia to industry is really pretty facile. And especially here in Cambridge, I just saw the Merck research labs as I was coming up in the Uber, and there are plenty of them around, so it's not hard.
Absolutely. Yeah. And when you did get your start in biotech, you ended up staying. And you worked 5 years at Glaxo. Can you tell us a little bit about that experience?
Yeah, that was an interesting transition from cell tech to Glaxo. Glaxo had the Glaxo Institute of Molecular Biology in Geneva at the time, run by Jonathan Knowles. And I thought that one of the reasons I went there was because I wanted to find out more about how big pharma used biotechnology. I was an applied biotechnologist, if you like, and I wanted to find out so how do you use this technology in a pharmaceutical company? And it was interesting because they were a little bit, I wouldn't say behind exactly, but they were using cloning technologies, which is what I was mostly involved with, for slightly different reasons. They were using it to set up screens in yeast to look for new drugs based on color changes, and it was they weren't so much interested then in cloning genes to make proteins as products. They got into that a bit later, but the Glaxo Institute of Molecular Biology was doing that. A couple of their products, IL-2 for example, was one of the projects that they were working on. So they were doing some of that kind of biotechnology, but most of it was more to do with how do you use biotechnology in the context of what you might call chemistry-driven drug discovery, because it was a very chemistry-driven company, as you can appreciate back then. And but it was actually a huge amount of fun. There were some great people, and at that point in the GGR, as it was in Glaxo Group Research, there were some tremendously talented people. The neuropharmacology group in Ware, for example, was known in the country as being almost the best, along with Merck, best neuropharmacology departments in the country. They were as good, if not better, than most of the academic groups. In turn, they published papers, they found out things about new receptors that other groups had not done. So you're in a pretty effective frontline research environment, so you could learn a lot and help to contribute to the idea of making new drugs to treat people with.
Yeah. So that's very interesting, and you might have to help me out here a little bit because I'm not as familiar with the English biotech scene. But my understanding is that you mentioned cell tech is sort of a newer company. Glaxo is a very established big pharma. Was that a significant change of scenery for you? And, can you tell us about the differences in working in each of those positions?
Yeah, I guess GGR was a bit special because it was kind of isolated a bit from it wasn't that big then, because Glaxo was quite small. It hadn't joined with Wellcome, it hadn't joined with SmithKline.
Okay.
So it was, from the research perspective, I didn't find it that much different. We were doing different things, but the environment was kind of similar. It was a little bit more bureaucratic. It took a little bit more time to get things done. But given the level that I was at, it was called a functional director level. I wasn't quite senior enough to get involved in too much of the bureaucracy, but I was senior enough to have an influence over what was done. And Alan Baxter, who unfortunately died 5-6 years ago now, and I were quite influential. We had good relationships with the CEO, who was the ex-head of R&D. So we used to be able to have cups of tea with him and talk about what we were doing. So, you felt like you were connected to senior management, which was important.
So ultimately you end up serving in a variety of roles at multiple different companies over the course of your career. You mentioned that, the differences between working at Celltech or Glaxo at the time maybe were not all that dramatic, but was there a particular role that stood out to you as being one that you enjoyed? And then conversely, a role that maybe you felt you were most challenged in?
Before I came here, you mean? Before I came to the States? I don't know. I think my straightforward style, if you like, was a relatively apolitical way of behaving. Made me curious about how some of the decisions were made. And I'll give you an example, because it led to me leaving Glaxo to come to the US, actually. So I took a 3-month timeout from my job as Director of Biotechnology at Glaxo to write what was called the Glaxo Genetics Initiative, because genetics back then was the positional cloning was just starting. It was obviously going to be important for the industry, but it wasn't quite clear how. So I took a time out working with Peter Goodfellow and Sidney Brenner, actually, to write a short paper, basically, on how genetics is going to affect the industry and how Glaxo should embrace genetics and get into that in a big way. And I made the recommendation that they should form a group that would be 200 people in 3 years and, with a budget attached. And I presented that to the executive committee, the research executive committee, and they basically damned it with faint praise and said, wonderful idea, Tim. You can have 5 people this year and then 5 people next year in order to get it off the ground. And I said, that's completely not going to work. I mean, you're never going to get it done with that. In the meantime, Peter Goodfellow was a consultant for Sequana, based on the West Coast in San Diego, and they were obviously committed to raising money to do exactly what I was recommending that Glaxo should do. So it wasn't very hard for me to make the decision to say to my family, which was now 4 kids and my wife, that we're gonna up sticks and move to San Diego, get some good weather and have some fun, which we did. And it was good for them for being up for it. And we moved in 1993 in December, just before the earthquake, on December the 15th, actually, which we all felt.
Wow.
And came to San Diego. But the point of that story is that in the executive committee, when I was making the presentation, it was interesting to watch how some of my colleagues either supported it or didn't support it. There were people who said, budget's too big, we don't really need it, we can follow the usual course of business, which was fine. And there were people who were very supportive, said, yeah, we should really do this. But there were a group of people who were watching to see which way the decision was going to go. And then they decided to vote with what was going to work and not what was going to go down and not what was not going to go down. Because they wanted to make sure they didn't get covered in a painting that said,
Sure.
oh, they didn't really believe in this, or they really did believe in this. So that's what I meant by forthrightness. You have to believe in what you believe in, and you have to state it and let the cards fall where they may. You cannot sit on the fence if there's an important decision to make just for political reasons to keep your nose clean. That's not how I operated, and I always respected people who didn't operate like that. It's not for you, it's for what you believe in and what you think is good for the organization. And it may or may not work. They may say no, which is fine, and you can either say go with the no, or you can take an alternative path, which is what I decided to do. And I had a meeting actually with one of the senior guys at Glaxo who had the temerity to say to me, and he was a knighted guy, right? He was a Sir this, I can't remember his name now, but it doesn't matter. He told me, did I really appreciate what was going on at Glaxo and that I should stick around because there's lots of good things to do? And he had the temerity to say to me, you're moving your wife and your family to San Diego to work for some cowboy outfit out on the West Coast. And I thought, what do you know? I mean, this is a ridiculous thing for you to say. So, what was I, 30? What was I, 43? And I thought, come on, guys, this isn't going to work. So that was actually another reason for wanting to leave, to start something special in San Diego. And I can tell you, Sequana was special. It was an amazing place, amazing atmosphere, really good people, and an amazing scientific advisory board. Probably the best scientific advisory board of any company ever, in terms of the key geneticists that were in academia wanted to join up with us and help to make Sequana successful, which it was to an extent.
Yeah. Wow. I can imagine, that attitude can be a great strength, but also it maybe ruffles some of the feathers of your superiors occasionally.
Tell me about it.
Right.
It definitely does. But you learn, I guess, over the years. You can do it in a polite way and you can do it in a less polite way. And I got more polite as time went by, I guess.
Yeah. And so we want to talk a little bit about your book, In Pursuit of Unicorns. But I wonder if you could just sort of wrap up your story about transition from England to the U.S. with a little bit more on Sequana and that scientific advisory board that you said was special.
It was a special place. And we used to have scientific advisory board meetings at Sequana. People would fly in from the UK and from other parts of the US. And we used to go for a run down to Flat Rock. And if you've ever been in San Diego in La Jolla, you'll know where Flat Rock is. You have to go into the park and then down to Flat Rock and up the other side and around. And so we used to do that. And when we started running, used to do it in the morning, everybody would be together and they'd be talking, and we'd be talking about what went on at the Scientific Advisory Board, what we wanted to talk about the next day, and it was all very jovial and kind of lots of communication. So you get down to Flat Rock and then you start going up the hill the other side, and then the conversation would gradually tail off and be clear that somebody will begin to run a little faster than the other guys, and then other people run a little faster to try to keep up. And gradually you'd end up at the top where you were going pretty quickly, and there was clearly somebody who wanted to win, even though it wasn't a race. And that gives you an idea of the competitive feel of the whole organization. Yeah. Everything was done like that. Everything was a competition. And,
wow.
My wife often says to me, it isn't a competition what we're doing today. We're just doing it. You don't have to win, right? But part of the passion for the science is the passion to win, and it can be little wins. It doesn't need to be big wins. You don't have to, and winning doesn't mean you write your name on the wall that I won. Winning is in your head, right? And it's to help you keep motivated to do what it is that you want to do. Does that help you in understanding a little bit more about Sequana and the atmosphere? And everybody was like that. We were all in it to win for ourselves and for the company. But as I said, not to write it on the walls, but just for the personal gratification of having won whatever battle it was that you thought we were in.
Yeah, absolutely. I think that sort of culture is definitely a defining attribute of many great successful organizations, whether that's in industry or in academia, is that sort of competitiveness in science. It's a very healthy part of the scientific enterprise.
And I hope it doesn't disappear. I worry about the present environment and the way, the rise of Chinese biotech. My mates tell me if you go to China, that attitude is exactly what you find.
I see.
You find that kind of competitive attitude. They all want to win. They all want to get the clinical trial started with their new molecule, and away you go. And I think that's got lost a little bit. And it's not the fault of biotech in the US. It's the fault of the administration that we're currently in. It doesn't lead to that kind of feeling. And we want to get the youngsters into the organizations in order to continue to do that. And there are some, of course.
Just a question, like how does the competitiveness align with the collaborative nature of like a big company, like where competitive and collaboration start, or vice versa? What are the main attributes of a successful company? One is competitiveness. You want to win. You want to do your best. And what else? I think it's
a team game. You have to understand that there's the bigger team. So you watch a football game. We'll use your football this time, not my football. And the guy scores a touchdown, right? Well, that's great for him. But actually that touchdown is great for the team because they've now scored, 7 points if they get the extra point. So he's got his win. The team has got their win because they've now got 7 points on the board. And I think you've got to look at the sports analogies are pretty strong. Those sports that require teamwork require the collaboration, but then you have the competitive because you've got the other team on the other side of the field. So that's the sort of sport that is if you like, my sort of sport. I like rugby. I like those kinds of team games. I've never been particularly into individual sports because I think some individual sports are rather egotistical. I mean, golf is a great example. I have great respect for the skills, but why do they get off on doing that? It doesn't, because there's no, unless it's a Ryder Cup, there's no teamwork involved. And then there's a Ryder Cup and it's a whole different story, as you know from a few months ago.
And how would you say did the culture change when you moved from the UK? How is the culture different in the biotech industry from in the UK to the US? Do you see a lot of differences?
Yes, it is less formal. It certainly was less formal back then. I think now, I mean, there's lots of less formality in the UK, but I think one thing you notice about coming here from the UK is how much less of a formal place it is, particularly the West Coast. The West Coast is less formal than the East Coast, which is one of the reasons why I guess I feel more comfortable on the East Coast than the West Coast. But I think that level of informality is great because there's a bit of looseness to it, the looseness to the teams, but there's still clearly team building and team working. And you go to any company, one of the things that you learn, and with the mission and vision statements and all the things about the way the company operates, is the team is the most important thing. Let's get the team working effectively to do whatever it is that we need to do. And you can have your personal wins in your head, but the biggest wins are the wins for the company.
That's cool, yeah. So we're just going to shift gears a little bit and talk about your book now, which was the In Pursuit of Unicorns, which was an incredible book. And we have Fang here, who's a big fan, and he says that the Amazon rating was a complete farce, and it was a great book, and everyone should read it. Whoever's listening should read the book. It's incredible. And the reason it's incredible is because unlike a lot of the biotech histories that we read on paper, it's usually written by journalists, but this was a perspective from someone who has actually helped build the industry from the inside. And it kind of gives a reader like a sweeping view of biotech's evolution from the beginnings of like recombinant DNA technology to today's cutting edge science. And CRISPR, CAR-T, mRNA vaccines, and it's interesting because it doesn't really focus on a single company. It basically gives you a wide range of how different companies work and how have they contributed to today's biotech industry. And so, like, the first question is, what motivated you to actually write the book, and what did you actually want most readers, especially young scientists like us, to take away from it?
Yeah, well, a couple of things. The inspiration was partly my father, who had written a chemistry book about carcinogens, actually, and I remember him doing that. The other inspiration, if you like, was that I didn't want to write a book that was just about one company. There was books about Genentech, books about Vertex. You can read about one company. What I was interested in was mapping the history of the industry from recombinant DNA to the present day, and do it from the perspective of the companies that were formed around the technologies that I was pretty well aware of, because I thought of myself as a technologist, which I primarily was, and I was interested in to record those companies that have been started based on particular technologies, whether it be positional cloning or whether it be siRNA or antisense oligonucleotides or messenger RNA or antibodies or recombinant DNA. And I'd read plenty of books about individual technologies, and I'd read books about individual companies, but I'd never read a book that actually took us on a journey from the beginning to where we are today. So I wanted to write a book which was really about the companies that were formed as a result of the technologies that were developed primarily in academia and had been subjected to several Nobel Prizes, each individual technology, which ended up being a company or more than one company. It's interesting how many of them actually ended up with the founders being Nobel Prize winners. So I was intrigued by that bit. And so I wanted to make it a journey, if you like, from the beginning to where we are now. But I didn't want to make it too much about me. I wanted to make it about the technology. And the way that I decided to write it was to say, well, it's my journey, and I'll talk about my journey, but at the same time, I'll bring in all the other companies that were relevant to my journey. So if we're talking about Sequana, you talk about Millennium, because they were a company set up at the same time. And there are several others in the book who were also pursuing positional cloning. So that's an example. And there were Sirix was doing structural genomics was doing structural genomics. So I wanted to cover the technologies at the same time as taking people on the journey that I'd had from the beginning all the way through to the end. And the reason I did that was it's easy to write anecdotes when they're personal anecdotes. So I went to Celtech and there was no distilled water to begin with. So where did I go to get the distilled water? I went down to the local garage to get the water they use for car batteries, because that's distilled water. And I could run my gels in the first couple of weeks because I had some distilled water. And I wasn't alone in that, as you read in the book. There were other companies that did very similar things. They went across the border to get their alcohol because there was no alcohol that they could get because they had no alcohol license. So the way that startups think about the world, and the way that you have to be pragmatic and you have to be creative in the way that you overcome the issues, I think was important, and I tried to wend that into the story.
That's interesting. So how would you say the startup, in terms of your thinking, differ from like the big companies that you ended up joining later on?
Well, because they're scrappy, right? You have to be there. You have to do. You mustn't be shy if you're the CEO of a company of sticking stamps on letters. If that's what you have to do, then that's what you do. And I think one of the attributes of the people who started various companies, Genzyme and others, is that the CEOs were not shy at the beginning in living in an uncomfortable office in an old chair with an old table, sticking stamps on letters in order to get the company off the ground. And then once the company was off the ground and they had their new building, they could build their opulent office with a lovely view over the Charles River. They could do that then, but they started in a very scrappy way. You mustn't be too pleased with yourself. If you want to do a startup, you have to do what you have to do, and then you can be semi-pleased with yourself once you've built a successful company. But you must never be too pleased with yourself. That doesn't work.
So writing a story like this is incredibly ambitious, obviously. So you mentioned, so how long did it actually take you to write the book? And like, was it just you sitting in a corner and just writing it? How did you decide when was the right time that you should start writing it? And what was the process like?
Well, it's partly COVID, because we were all hidden away. I didn't actually start writing it until, if you like, the second year of COVID after being vaccinated. But not that just meant I could go out, but not. But it wasn't particularly slow because I was on Zoom quite a lot, but I had time to think about what it is I wanted to write. And the most important thing was, okay, so what am I going to talk about? What are the chapters going to be? And what am I going to cover? And once you decided what you wanted to cover, it was relatively easy to construct the narrative in the chapter. So recombinant DNA, pretty straightforward. Monoclonal antibodies, you know where they started, you know where they went. I could cover the products, I could cover Keytruda, Humira, things that were important to the industry. That was relatively straightforward. And as you got more current, then it was easier because you didn't have to go back to the 80s to get the information. You could get the information pretty directly. And one of the things that I've always done is to be pretty up to date with what's going on. So I read Science and Nature every week. So it wasn't difficult for me to recall, for example, CAR T papers. And if I didn't know, I knew who the authors were, so I could go find them, go read them. I would get to understand a bit more about the origins of the companies that they started. So it was a mixture of what was in my head coupled with what I'd had on my computer in terms of the necessary papers that I needed to read. And then you could go get the information using, the current, by Googling it essentially, or actually, interestingly enough, Yahoo was often a better source of older information than Google was, because you can get for older information by Yahoo searches than you can on Google, as I found out. So if you wanted to get, for example, a letter that you thought you knew about and you wanted to find it, you put that into Google. These days you get nothing but a stupid Gemini summary, which is often wrong.
Right.
Now you can, back then, even when I was writing it 3 years ago, you go to Yahoo, you would probably find a reference to the letter, and then you could go find it. It was that, and I talked to a lot of my colleagues, as you know from the acknowledgements. So, like Immunex, I know Doug Williams pretty well, so I asked him about Immunex and how that all started, and I know Chris Henney with ICOS, so it wasn't hard to get the information about the beginnings of the companies. Some I could do. Stelios Papadopoulos, who was an investment banker with Cowen, knows all the companies, has S1s for the IPOs of every company in the biotech industry that has ever gone public. So if you want information about when they went public, how much did they raise, it wasn't hard to ask him, and I could get the necessary information. So I used my network, I used the search that you need to use, different mediums, as I said, and I had a lot of information of my own that I could put into it. And it probably took 20 months to write, and then it probably took, with my fabulous editor, Finton, it took probably 4 months to edit it. We did a chapter a week, pretty much. I would send him the chapter. He would put his comments in. I would get it back. I would edit his edits and then send it back. So we did it chapter by chapter. It wasn't very hard. Most of the key things he did was to put things in the boxes, because I have boxes about some of the technology. Some things I didn't put in, he put in, and some of the things that he put in, I took out. And so to try to keep the narrative flowing without having the reader have a lot of technical stuff that they need to read. They don't have to read what's in the box about the technology. They can just continue to read, right? So if you want to know about Talens or zinc fingers, you don't have to read about it in the text. You can read it in the box if you want to.
And how did you decide on the title, The Pursuit of Unicorn?
Well, that's an interesting story. As you know, unicorns are billion-dollar companies, right? Mm-hmm. And I talked about in the book about the origins of many unicorns. And I thought, well, we've got to have unicorns in the title somewhere. So I was sitting with my offspring, my son actually, and said, it's about unicorns, what do you think? And I think we were hunting unicorns and we thought that was a bit too strong. So either they or I came up with In Pursuit of Unicorns. And then it had to have a colon, and then what about a journey through 50 years of biotechnology? So that's how the title came up. And then Finton, my editor, thought unicorns was great, and he said, have you thought about the tapestries that hang in the cloisters in Manhattan, which are, I think 16th century Dutch tapestries with unicorns on them? And I'd actually never seen them, never heard of them, in fact. It shows how ignorant I am of the arts. And but I looked at them and I thought they would make fabulous cover for the book. So it turns out they're freely available, you can use them. So Cold Spring Harbor Laboratory Press, who published the book kindly, put it on the cover, and you went from there. And it's astonishing how stunning the cover of the book is, and everybody says cover's stunning, and I said, don't judge the book by the cover. Please read it, because I hope the book is as good as the cover. But the cover art was great, and it's compelling, and actually the book is selling pretty well for a hardback. Talking to the guys at Cold Spring Harbor Press, it was the best-selling hardback book of last year, and as a result they wanted to do a paperback edition, which is coming out in a couple of months, or maybe a month even.
Great.
And it has a new chapter 19, which I can tell you about if you want to hear about it.
Please.
Yeah, for sure,
yeah.
please.
Okay, well, chapter 19 is called Dystopian Times, and it actually brings the readers up to date with some of the companies and some of the technologies that I talk about in the book. So, for example, I talk about Sarepta as an example of a gene therapy company that has gone through some rough times. So I elaborate why and how, and maybe there are some lessons to learn from how Sarepta managed their interactions with the FDA. I talk about the rise of Chinese biotech because that's definitely different now to how it was when I wrote the book. I talk about artificial intelligence and how many companies have been started based on AI and how much that is more of a myth than a reality, in the sense that the AI is not a myth, but the way that it influences drug discovery and what they say that we will discover you a drug in 18 months is a myth, because drug discovery is an experimental science. They might find you starting points more quickly, but you still have to do the experiments to sort out one thing for another. So I'm a believer in AI in the sense that I think it helps you to find your starting points, but then as I put it, then the fun starts. You still have to do what I think is fun, but it actually takes time to elaborate what it is that you found using artificial intelligence. And there are lots of very intelligent people using AI to help them to do their drug discovery activities. So that forms a reasonable proportion of Chapter 19. And I do go into the politics of the current environment and how difficult it is with the FDA and how difficult it is for some of the academic institutions like this one to manage in the current environment. And some of my guys tell me it's actually a bit easier now than it was 6 months ago, but it's still pretty tough. And that is going to take a lot of time to sort out. And my worry is that when you have the rise of Chinese biotech and you have the demise, if you like, of US science driven by a very unsympathetic government to what it is that we do for a living, I think that's not a very good recipe, and we'll end up buying our drugs from China rather than buying them from the US, which is what we're used to doing. That will have long-term ramifications. It's not, at the end of the world. It just changes the equation quite a lot, in my opinion.
Absolutely. It's definitely a challenging time for biotech, but of course, as I'm sure you're aware, biotech has been through its share of ups and downs. It's a very volatile industry, and so it's faced challenges before. Can you comment on how this may or may not be a different challenge that the industry is facing?
Well, I think that you would, given the current state of the markets, for example, and often biotech investment is driven by the general market position, the markets are not doing so badly, funnily enough. So in a way, the industry is being protected by the lack of volatility in the markets. The markets are generally going up in the usual sort of way, a little bit of up and down along the way, but the biotechnology index has certainly gone up in the last 6 months, which I think helps investor confidence. I understand that the JP Morgan, which is obviously the big biotech conference which is held in January in San Francisco, was pretty positive this year, and there is the expectation of more mergers, so more acquisitions by big pharma of small companies, which already drives investment, as well as the opportunity for 1 or 2 IPOs. So Pearl Mutter's company is filed for an IPO this year. There are other companies who are getting ready to file if that should go well. So we'll have to wait and see. But I think it's not being influenced quite as much as I thought it would be from that perspective. But nevertheless, there is the challenge, if you like, of the Chinese biotech scene and the focus of the industry right now in accessing clinical stage assets from China and building companies based on those assets that they've got from Chinese sources. I think that is fine, but it mustn't be at the expense of innovation that starts new companies here in the US, where they got a new technology that they want to develop new ways of discovering drugs. And I think it's still the case that innovation happens here. It happens to a certain extent in the UK. It happens less in China, but it's going to increase as they get going. When I did my PhD, 1 in every 500 papers that I read was from a Chinese source. Now it's probably 5 out of 10, 8 out of 10. You look at Cell or Nature or Science. It's amazing, actually, and they're pretty talented people and very well informed, and they publish some great papers, so I'm not knocking it. It's just different.
Right. Yeah, so I hate to be sort of pessimistic, and you've given us a few reasons to be optimistic about the state of the biotech industry, but is it possible that, it just takes time for us to maybe start feeling some of the ramifications of these policy changes in the U.S. or what's, been happening overseas? Of course, it takes a long time to translate basic science into a therapeutic under ordinary circumstances. So if we have this sort of lack of investment at the beginning of that pipeline, it might be years, if not a decade, before we're seeing sort of drop off in the production from our scientific pipeline.
I think there will be an effect, and I think things will slow down in that respect. But I don't think it's going to slow down to such an extent that it kind of kills the business. It won't kill the business. There's always a way around it, and the creativity of the people involved will ensure that happens. The situation, I guess the thing that worries me most is the situation at HHS and the lack of scientifically driven decisions. You can see that in their vaccine policies. You can see that Kennedy surrounds himself by people who don't know any more science than he does. And that is a problem because the industry, as I've outlined in the book, has been driven by people who have a fundamental understanding of science and how you can use it to do things that are going to help other people. And there's no better chapter to read about that than the vaccine chapter and the whole messenger RNA piece from Moderna and BioNTech. They didn't discover messenger RNA and its potential for vaccines, but they were great at turning that discovery into a real product. And if it hadn't been for them, we would be a whole lot worse off than we were. And my worry is that should that come round again, is there another technology that is ready and waiting and would be supported by the present administration? And the answer to that question is there isn't. And that is what worries me, particularly in the vaccine space. But it kind of worries me elsewhere as well, because we need scientifically driven decisions and not politically driven decisions when we're making decisions about what drugs we should give to people.
Absolutely. And we do have the book here in front of us. You're right, the cover art is beautiful, but there's a lot of wisdom contained within the book itself. So, of course, we encourage our listeners to give it a read.
So there's an equation in the book that wisdom equals experience times smarts, right? You can't change smarts. You're as smart as you are, but you can change experience. And so wisdom equals experience times smarts. And you say there's a lot of wisdom in the book. I thank you for that, because wisdom is an important thing to have. And it's not something that you find in very young people. They say, oh, a very wise young boy, right? Nonsense. Wisdom is something that you gain as you gain knowledge, and you couple that with your ability to integrate data and explain it. You can argue with me about that if you want to, but because other people have definitions of wisdom, but I think that's definition of wisdom. And the wisest person I've ever met is Sidney Brenner. He was amazing. The wisest guy I've ever met. Not only really clever, but a huge database and lots of common sense. And when you mix that in, you get a pretty wise person.
Yeah, I think genuinely there is a lot of wisdom in the book. And as you say, if wisdom is sort of your smarts times your experience, that wisdom comes from all of your experience in the industry over many decades. But what's interesting, for me at least, is how many of these lessons in this book are still relevant today. And, as you've kind of been commenting on, there's a lot that can be applied to the current climate in academia and in the biotech industry with what's been going on sort of politically. And, I've heard you say that in some sense the next generation is the generation that's going to be tasked with maybe charting a new course in some ways. I was hoping you could maybe distill some of the wisdom that you've included in this book to sort of give to the trainees that may be listening to the podcast, but particularly those that are hoping to break into this industry, either as founders or eventually as CEOs and the next generation of leaders?
Well, I think my answer would be, don't be afraid to follow your passion. If you have a passion for a particular technology, you have a passion to achieve something, then don't be frightened of doing what you need to do to follow that passion. Risk is what are the risks associated with that? If you spend too much time trying to analyze the risk, you'll probably end up not doing it, because you'll be in a situation where you think, oh, don't want to do that, it's pretty risky. But life is short, and if you really want to do it, then do it. And it's the same in science as it is in music or in the arts. If you really want to do something, do it. Find a way to do it. I watched a film about Eric Clapton last night, actually, because he's one of my heroes, because he's such a great guitarist. And it's really obvious right from the very beginning that he followed his passion. He wanted to play the guitar. That was it. He wanted to play the blues, so he did. And not everybody has the talent to be able to succeed, but don't give up because you think it's too risky. You've got to take the risk and see where it goes. And there's always a way to move forward. Never give up. Just find a way to follow your passion. And I think I was fortunate because I knew what I wanted to do when I was about 16. There are many people who don't know quite what they want to do, and you have to have little other experiences in order to find out what you're really passionate about. But that passion is really important. Follow what you really want to do, because you'll do it well. It's inevitable. If you spend a lot of time doing it, you'll get to be quite good at it. The 10,000 hours Michael Gladwell or whatever it was rule, I think it's true. I believe in it. And I've spent more than 10,000 hours in biotechnology, so I hope I'm reasonably good at it.
Yeah, going back to what you said before and how the landscape's changed in the biotech industry, with so many changes, what kind of lessons do you think we're at risk of forgetting from the previous generation that we could apply to this new generation of biotech, and how would the past inform our decisions now?
Patience with a C as well as with a T. You have to be patient. You have to have persistence. You really have to continue to do your thing, because there'll be lots of humps in the road and you can't give up. And one of the things that you notice about the companies that I've written about is that there are some, Regeneron, a great example, where their first products didn't work. Fortunately, they had some things on the back burner that they could bring to the front burner that did help them, and they're a hugely successful company now, thanks to the leadership of that company. And Alnylam, another great example, John Maraganore talked about it on this same podcast. They went through some pretty tough times, and they ended up being what they are because they had persistence, and they coupled that persistence with a vision of what they wanted to be, and they had a realistic view of what they had to do to get there. Same applies to Millennium. They realized that they weren't going to make it just by using genomics. They had to find products, which is what they ended up doing. So you have to be practical, you have to be persistent, and you have to have patience. So it's the 3 Ps really.
But with the current competition in the biotech sphere, there's so many companies coming out. Is patience actually possible?
Yes. Patience if you go to the right place. So if you're going to join a biotech startup, you have to choose the right one. They have to be well-funded, or have the prospect of being well-funded. They have to be well-run. You have to follow the no assholes policy. You can't operate when you have people that you don't trust. You have to be in a situation where you can go for a beer with the people you're working with and actually enjoy it. That's really important. So it makes it easier to make decisions, because they're not necessarily friends, but they're good colleagues, and it's a little, as I say, it's a team game. It's like playing football. The football players aren't friends with all of their other players. They're friends with 1 or 2 of them, but they work with the other colleagues and they go for a beer and things. So it's not dissimilar. And it's a good analogy, actually. I think the sports analogy is really strong. And, I watch a lot of sport because I like it, and I like the attitude of mind that goes with it. And I've got strong views about which teams I support and which teams I don't, right?
So do you actually encourage, in the current climate, you actually encourage graduate students or maybe postdocs to actively spin out, make their own companies, perhaps?
If they have technology that they're passionate about doing, absolutely. Do it. Try and find the money. Don't give up. There's plenty of VCs out there who are looking for new things to invest in. You just have to couple yourself with some advisors who can help you push you in a particular direction, and people who are not interested in necessarily being paid for their services, at least to begin with. You have to do it because you want to help them, and then we'll see where it goes. And I've done plenty of that, actually. Not always successfully, but sometimes.
So looking forward, which areas do you think will give rise to the next biotech unicorns? And where do you think we should place our bets, basically?
That's interesting. Properly run organizations that use AI effectively to help them in the drug discovery process is certainly a new way of going forward. So AI will have a role, but it will be a very selective role and a very thoughtful role. It's not going to be an AI company that is just going to do computational run after computational run. It's got to have people who understand the drug discovery process and move forward with that. So that's one area. The other area, and I listened to a company this morning actually talking about it, are molecular glues. These molecular glues are really clever, where you can effectively change the specificity of a ubiquitin ligase to degrade particular proteins selectively. I think that is going to be a really important technology because it uses chemistry to find small molecules that interfere or affect protein-protein interactions. So it's basically it's a PROTAC derivative. So PROTACs are difficult because they're big and they're difficult to find and they're difficult to make. But these small molecules are just normal small molecules that you can look at using structural biology. And as you know, the whole structural biology scene has changed. With cryo-EM, you can get structural images of small molecules attached to big ones pretty quickly. And the best, as you know, the best AI tools are available in the protein prediction sphere because there's more good data available on which to build the algorithms. And that's the problem with AI when you're thinking about cell biology or you're thinking about drugs in people. The databases just aren't there to help people do what they need to do. The beautiful thing about protein structure determination is the databases are full of protein structures that have been determined over the years, and the algorithms are pretty good at predicting what's going to bind to what or what structure of a protein might be.
Yeah, it's very interesting to think about, these new ideas and specifically investing in these sort of the future of biotech. And I think you mentioned that you're now on sort of the darker side of the dark side of the industry. But could you tell us a little bit about what you're doing now?
Yeah, I spend a couple of days a week working with SV Health Investors, and I'm primarily technical due diligence guy. So, for example, I listened to this glue company this morning, and I could understand what it is they're trying to do. I could appreciate what they can do that other things can't do, and I can tell my colleagues at SV, having listened to it with a couple of other colleagues from SV, that this is something that's pretty exciting, and maybe we should think about how we might organize to help to invest in this it's a new company. It hasn't started yet, really. It's a spin out, and I thought that's the kind of best example of what I do. If you like, some of the things that, if you like, get in the way is that it takes time to do the due diligence, and sometimes the due diligence takes more time than you think it should. If people would read the answers to the questions that you just asked the company, then you probably have got answers to most of your due diligence questions. You don't need to have yet another conversation with them to go through the same thing. So there are VCs that spend more time doing diligence than others. But the most important thing about what I do that I get most excited about is enormously science-driven. Most of the decisions that we make are driven by the quality of the science, the quality of the team doing the science in the startup, and the quality of the management. Are they good? Are they not assholes? That's the no assholes rule is really important. Does the team work together? So if you have a CEO that spends all his time talking about the company, he's got a group of his colleagues on the Zoom call and he doesn't involve them at all, that's a signal, right? If you've got all your colleagues there, why are they there if you're not going to use them, right? So I tell my people who I work with, who are CEOs, look, if you're going to answer the question, have one of your guys answer the question, or have one of your guys make some of the presentation, which is something that they know about. Don't try to do it all yourself, because it doesn't sit well sometimes. You're not the only guy. You might be the leader, but you've got to use your team. So that's a recurring theme here, I think, is teamwork is really important, even if it's only a 3-person startup. The way the team operates and how they work together is important.
Yeah, absolutely. That's very interesting. The VC world is absolutely essential to, driving innovation, and it's always interesting to hear, as science-driven as a lot of what we do is, the intangibles are also, incredibly important. And you mentioned, doing due diligence and, companies that are sort of harnessing AI in various ways. Perhaps like diligence is one of those areas in which AI can make things more efficient. You've touched on this a little bit already in talking about the ways companies are harnessing AI, but are there any particular areas that if you had sort of unlimited resources to invest in, you would sort of put your resources there?
If you had all the money in the world and and you didn't have any regulations holding you back.
I think it's a data-driven business. AI. You've got to have quality data in order to draw the appropriate conclusions. And I think there's lots of clinical data about the effect of small molecules or larger molecules on particular diseases, but capturing that information in a form where you can use algorithms to distill out of that information what is going to work and what has not worked, so that you can reduce that time to get to the right molecule, I think is going to be important. And so one of the bits of AI that needs to be invested in is making sure the data sets that you're going to use use your algorithms on are well documented, and that its accuracy in the data that you are examining, because it's a crap in, crap out situation. And I don't think some of the people, particularly the really heavyweight computational scientists, appreciate the drug discovery business and the quality of the data that they're analyzing. As I said, protein structure information is the best source, but there are other data sets that we need to be able to harness and look at carefully as a starting point. So that's what I would do if I had if you want to use AI in all aspects, then you've got to have the clinical piece to go with it. That will save some time, because then you'll do your clinical trial in the most appropriate people. And one of the time-consuming bits is getting the right people into the clinical trial. That takes a long time and quite a lot of money.
So you would invest in drug discovery if you had all the money in the world?
Yeah, of course, because that's what I do, right? I help people to do. I don't do that myself. But I've built companies that do that and worked for companies that do that. That's the exciting bit. I mean, watching, particularly in the monoclonal antibody sphere, this is one of my pet peeves. You listen to the news on the television and every advert is an advert for some monoclonal antibody. It pisses me off, because I don't think you should advertise pharmaceuticals on the television. I've said that in the book, and I've said it in chapter 19 again. Nevertheless, they do. And but if you look at them on the face of it, then it just shows you how incredibly important the industry has been for people with psoriasis, people with ulcerative colitis. All the things that you see advertised is a testament to what the industry has delivered. So if you look at it from that perspective, then I'm not so concerned. And the adverts that I like best, if I like any pharmaceutical adverts, are the ones that Merck tend to do, which is more about Merck being a drug discovery organization than advertising a particular product, although they do that, not to the same extent as AbbVie, for example, which I think overdoes it.
And then I guess on the topic of drug discovery, there is a huge debate in the country right now with how high the prices are for drugs and how unaffordable they are in this country, and how it's not how these drugs are not actually getting to the people that actually need it. What would be your comment on that from the other side of things?
Well, it's tough because you can look at it from several perspectives. It's an expensive business discovering drugs, there's no question about that. And I think the US view that we do most of the discovery and we set the prices, and you in Europe sell our drugs for a lot less than we sell them for, is a fair comment. Nevertheless, the solution to that is not necessarily it's two things. It's one, reducing prices here, but it's also increasing prices where there are people taking expensive drugs at not the same price that perhaps you would anticipate they ought to be paying. I'm glad they're getting them, but they ought to pay. The governments should pay what it takes. And actually the UK is one of the worst offenders in that respect. They don't pay what they should pay. And I think it's a question of how you do your maths. If you do your maths and say, well, the price of the drug is $200,000, then that's one thing. But if you realize that by paying the $200,000, you take the patient out of hospital 5 times a year, and they go in for 2 or 3 days in a acutely sick or chronically sick, the amount of money that you save by treating them with a $200,000 drug may be way less than what it costs in the year that they have in hospital because they didn't get the drug. So you understand what I mean? That's a bigger calculation or calculus than I think people, they look at it from the perspective of the price of the drug, but you've got to look at it from the perspective of what does that price buy you. It doesn't just buy you the drug, but it buys you other things as well. Like, I'm not going into hospital, I get a longer life, I can go back to work earlier, all the kinds of things that are, if you like, hidden, but they're important in the economic analysis. And I'm absolutely no expert, but I appreciate what has to go into the calculation. What comes out the other end is not for me to say, but it's not simple.
Definitely not. And it seems like there's just an increasing number of new ways in which we're distributing drugs. The sort of increase in the direct-to-consumer marketing that you mentioned is just sort of one example of how companies are adopting different strategies. There's also these sort of now private access cohorts of physicians in a new format in which you pay a small fee to participate in this sort of small group that you have a more direct access to physicians and things like that. So it's interesting how that landscape, especially recently, is evolving in response to this sort of debate. And sure, go ahead.
No, I was going to say I don't really know very much about that, but philosophically, I think there should be equal access for everybody. You shouldn't be able to, if you like, pay your way to a drug that somebody who can't pay their way to the drug can't get. There has to be equal access. It's up to the physicians to make sure that the patients that they're seeing are able to get the drugs that they think they should get. So there's a lot of physician responsibility here to make sure that it's even-handed, and that, I could afford to buy whatever I needed to buy from a drug perspective, and I don't mind doing that, but other people need to be able to have access to that drug as well, even if they don't have the financial resources that I have. That's a philosophical statement.
Absolutely.
And it's a political statement too, in a way. That's how I think things should run. There are groups of people who make money, and there are groups of people who don't have any, and the people who make money should make sure the people who don't have any get looked after.
So we're running a little short on time, but before you go, I was hoping you could just share with us, maybe a few of your hobbies. I know you mentioned that you have some strong opinions about the sports teams that you support.
Yeah, hobbies. What do I do as hobbies? Well, I work. That's kind of my hobby. Work-life balance. Well, it's been a tough thing for me over the years, and my family remind me constantly about that. But you look at John Maraganori's same deal, right? What does he spend his time doing? He spends his time building companies. That's kind of, once you're in it, and it's difficult to not do it. There was a time when all the beds in my house I made. Doesn't mean to say that I'm a particularly good carpenter, but I do know how to follow a carpentry recipe. And I built bunk beds for the kids, for example. And I really enjoyed doing that. I haven't done much since.
Cool.
And I met somebody who does it professionally, if you like, even though they're an academic scientist. And they're so much better than I was. You think, oh shit, I wish I could get as good as that. But I've never got there. I played the guitar badly when I was a kid. I played a couple of folk clubs when I was a teenager, and I'd like to play the guitar better, but I never had much time to do that. If you like, that's a hobby. I like to get out in the garden and just do things as silly as raking leaves up or planting vegetables. I kind of like to be with my feet firmly on terra firma, looking up at the sky and just being here, right? Yeah. And I think you can do that if you get out and about. So I watch people doing kite surfing in Nahant where I live, and you think, God, I'd love to have done that when I was a kid. And I spent time in San Diego and everybody was surfing, and I thinking maybe I should do that. But, I went there when I was 43 and I never got into it. I was too busy working. But there were guys who worked for the company who used to live in the garage underneath the facility in their cars, and they would have their surfboards on the roofs of their cars, and they would go and surf in the mornings. They would shower at work, and they would then sleep in the car. And it's just they didn't have money to didn't live in a place, they lived in their car. But they worked, and it's just back then that's the way it was. And funnily enough, that facility, 11099 North Torrey Pines Road, where Sequana was one of the first facilities at Alexandria. It was a big biotech real estate company. It was one of their first facilities. We were, I think, their second rental space in San Diego, because they started in San Diego. And that company now owns or runs probably 80% of the biotech space in the US. And I know the CEO because he helped me out in the course of money. I go into it in the book. One of the essences of biotech is location, location, location. And there are key areas, Seattle, San Diego, Boston, San Francisco. Yep. You know them. They're the places. And if you're going to start a company, don't start it in somewhere like Portland or because there's no biotech infrastructure. You got to go where there's infrastructure, because not only can you hire people easily, but all the VCs know the other VCs, and it's just much easier to be in a center of excellence. And there, as I said, I mentioned them. They are there, and there are one or two others, but they're not as important as the West Coast, San Diego, San Francisco, Seattle, and then here Boston, maybe DC, but not really. Maryland, you know what I mean?
Sure. Yep.
Yeah. Cool.
So, lesson is going to do a bite. Nothing is more exciting, I can tell you, than doing a startup and being in there. And nothing. I've started them and I've closed them, and I tell you, starting them is much more fun than closing them. But you kind of learn stuff along the way, and getting everybody together, getting started in a room maybe not much bigger than this, or getting around where are we going to get the money from, what are we going to do, who are we going to hire. Yeah. It's just fantastic fun.
Yeah, you've had a lot of valuable experience from start to finish in many ways, you've given us a lot of food for thought, and I'm sure we'll take away some practical lessons from this talk. So thank you for sharing your wisdom with us. We're really lucky to have you on the podcast.
And I appreciate it, and thank you for having me. And please, if you haven't read the book, read the book and enjoy it, and wait for the paperback edition, which is coming out shortly with chapter 19.
Absolutely. Thank you so much.
Thank you.
Hi everyone, that's the end of this week's edition of the GSAS Harvard Biotech Club's The Pulse podcast. A new edition of Tim Harris's book, In Pursuit of Unicorns, A Journey Through 50 Years of Biotechnology, is going to release on March 3rd. We highly recommend checking it out. I would like to thank Tanner for helping co-host this episode and Funk for producing it. Thank you to all the listeners, and stay tuned for our next episode.